Yes, sir.
Okay, thank you. So since the topic that was allotted, and this is centering around or in and around COVID, so I thought it is better to begin with the COVID pandemic. And if you look at today's fact, so it is December 4th in the morning, I downloaded the statistics. So these are the alarming figure across the globe, and this is from India. And currently we are the second runner-up in this race. And not to be proud of that, but since we are the second most populous country, possibly that is because we are on the second. So students, you must be now expert in all the COVID related biology, because this is around a year we are listening to this buzzword COVID-19, COVID-19. So this virus SARS-CoV-2, it is phylogenetically, that means with looking at the structural as well as genomically, it is similar to many SARS viruses that were earlier there, and where bat was one of the hosts. And we are still looking for any other intermediate host which we are in contact with. And now we are in this phase of human to human transmission, and struggling for getting a hold on this deadly virus. So if you look at the genome architecture of this virus, it is around 30 kilobase pairs, around 30,000 nucleotides. And that has got many structural gene as well as RNA dependent RNA polymerase and the spike proteins, all these. So we are now well aware of all those structural proteins which makes the crown of this virus, and it surrounds the genome and protect it. So it is a enveloped single positive stranded sense RNA with 29,811 base pair nucleotides. So these are the molecular facts, and these are the structural proteins which protects them. If you look at the life cycle of this coronavirus, first phase is once we come in contact with any transmitting or carrier, and our cells are exposed to that, then the first exposure is ACE2 receptor to which the virus binds. And the cleavage of this cell will make the entry of this virus into the system. And once it enters into the system, it hijacks our own translational machineries, and it keeps our epithelial cell busy in manufacturing its own structural protein as well as its own RNA and the nucleocapsid. And ultimately, it is being processed and it is thrown out of the cell in multiple copies, in thousands or in millions. And as a result of that, the cells which harbor them, they are lysed or they undergo apoptosis. And ultimately, it causes inflammation in the local cellular milieu where these processes are going on. And we are affected with various symptoms. Right? That means these are different phases of the viral replication cycle, and ultimately these are being released. So why we are looking at this? Because if you closely look at this viral replication cycle, you will find that which are the possible stages where we can play some role while we are designing some antiviral drugs, right? So whether we can restrict them from entering at the receptor level so that you develop some antagonist which goes and bind to the ACE2 and that doesn't allow the virus to sit over there. So that will be something which is prophylactic, right? So people says that you eat this and you will protect yourself from viral infection. So there are two ways. So either you prevent the entry of virus. So that we call it as prophylaxis, or we call it as chemoprevention. And the second one is, if it enters, can we restrict them at this level so that the viral genome is not released into circulation or into the cytoplasm? Or can we restrict them for their replication? So their enzymes, RDRP, which replicates the genome, can we restrict there? So all these red T, those are the point where we can inhibit. So the translation of viral replication machinery can be targeted. So here it is written as the CL protein or the PL protein, right? So these are the enzymes, helicase and all those, which can also be targeted. If not here, can we target where this viral structural protein are getting assembled? So these are viral structural proteins, or the entry of this viral RNA packaging into the structural protein, that stage can also be targeted. That means the viral assembly, or the exit of this virus. So exocytosis, or so at this point also can be restricted. So there are many opportunity starting from entry to exit, where as a biologist, or as a molecular biologist, or as a medicinal chemist, or as a pharmacologist, we can definitely play some role. And that is the current phase of research across the globe. So if you look at the literature, people are working on various aspects of this. So if you look at the first stage, that means whenever someone develops a mild symptom, so current list of symptoms are very long, but nowadays it has shifted to loss of smell, loss of taste, fever, all those. So if any of these symptom develops, we are prompt enough to call 104, or we go to the tents where these rapid antigen testing, RAT testing, is being carried out. And that is the process of simply collecting the specimen from the nasal cavity or the oral cavity. And the ultimate intention is to assess the presence of viral particle or the viral RNA. So if it is a rapid antigen testing, we are looking for the presence of viral protein in the specimen. And it takes hardly 10 to 15 minutes to develop that band. And if there is a positive band, they will declare you as positive and you are quarantined for 2 weeks. But many a times this sensitivity of RAT is very low. So if you get negative, that doesn't mean that you are not harboring the viruses. So now you are referred to go for RT-PCR. So as you all know, you must be aware of the reverse transcriptase polymerase chain reaction. So that is the chemistry behind that. So here, the first step is you isolate viral RNA from the specimen that you have collected in viral transport medium, VTM. And once the RNA is isolated, the first step is reverse transcription, and that's why it is RT. So this reverse transcription will make multiple copies of the viral RNA into complementary DNA, cDNA. And once the cDNA is ready, this cDNA is acting as the template on which the PCR reaction is carried out. And you all know that PCR reaction requires specific primers. So here, the primer they use 3, or many times it is 2, which is one of the ORF gene, RDRP gene, and some internal control to ensure that the person is having a copy of viral RNA or not. So at the end of this real-time PCR, again, you are assigned as positive or negative. And if positive, what is the CT value? So the CT value is the cycle threshold. So that shows that at which cycle the amplification curves come out of the baseline. So if the cycle value is less, that means the CT value is less, the viral load is more. And if the CT value is more, the viral load is less. So it ranges from 18 to 34. So if it is 18, it is highest. If it is 34, it is very mild. And based on that, your further treatment protocol can be planned. There is antibody test also that shows that if you have an infection of corona, say 2 weeks before, then usually it is better to ensure that whether you have developed antibody or not, or else there is a second chance of infection, secondary infection, or the second infection. And for that, now serological tests are available. It can be a simple pregnancy testing like strips, which is called as lateral flow assay, and where the anti-COVID antibody specific antibodies are being printed. And if you develop band at G or M, based on that, there is for sure that you will get this control band, otherwise the test is failed. If you get IgM, that means you are still in the phase where IgG is not developed, because IgG develops at a later stage, and IgM starts developing from day 7 to day 14 or up to day 21, and IgM will disappear and IgG will take over. So usually if you want to test yourself immune, it is better to test after 28 days, so that if you have got IgG, then you are lucky enough, fortunate enough that you developed the antibody against COVID. And now you are suitable to donate your blood for convalescent plasma therapy. So I think we already know a lot of these signs, symptoms, and diagnosis. So I'm not going much into detail of this. And finally, if you develop some severe symptoms, doctor will ask for a CT scan thorax screening, so that they will look for any patch, glass opacity in the lung. And that is a bad sign, because that impairs the lung function. If you look at the figures of incidence and death, right? So the death rate ranges from somewhere around 2% to 5%. And if you look at the daily death due to COVID, it is more than, say, 6,000 on an average. And the daily death of December 3 globally was around 12,683. Of yesterday's, right? So is this figure alarming? So if you look at the global figure of death due to any disease, then possibly cardiovascular disease is leading with somewhere around 48,000 deaths. And against this, if you look for this is around 12,000. So why we are so alarmed? Because this is currently actively communicable disease, whereas cardiovascular disease is very much individual specific, right? This can happen to anyone, a newborn child to a elderly man, right? So these are the issues. So when you roam around or in social media, there are a lot of fake news, myth, or these are circulated that you do this, and possibly if you can do this, you are free of corona. So few of these myths and facts have been released by WHO to make the people aware of that and don't get driven. That if you can hold your breath for 10 seconds, you are healthy. So everyone try to assess himself or herself and say, okay, I can hold up to 20 seconds, so I am fit. So these are all myths, right? And a few of these lists, these are recently being updated by WHO. And this says that vitamin and mineral supplement cannot cure COVID. That means if you take this as a prophylaxis, it is fine, just to maintain your healthy immune system, but it doesn't guarantee that taking these pills will help you in curing COVID, because these are not directly antiviral per se, but they definitely help you to maintain a healthy immune system. Hydroxychloroquine doesn't reduce mortality of hospitalized COVID-19 patients. Adding pepper to your soup doesn't prevent you from COVID, right? So these are few of the facts which need to be transmitted to the general mass. So then why people die of COVID? So that is because it is a lack of immunity in them. So around 5% of the people had severe lung injury and even multi-organ dysfunction, including respiratory failure, cardiac failure, viral sepsis, cytokine storm, and the fatality rate is somewhere between 1 to 5%. And it is variable from country to country and from the developing to developed country based on their facility and all those. And it has been found that this immune response against COVID is biphasic. That means the first phase, when there is sudden exposure of the virus as an antigen, which is new to the human body. So it is a protective response, right? So it is requirement of T cell, that means T helper cell, activated CD4, CD8, and the B cell, which is required to release the immunoglobulins. Whereas few of them who enters into the phase 2, and this phase 2 is the damaging. So our own immune system is unnecessarily being activated to a point where it enters into cytokine storm. So you must have heard that after a certain stage, people needs admission and then ICU care and ventilation, and ultimately there is cytokine storm and a lot of various interleukins and cytokines. They make the life miserable, and ultimately the person succumb to death. So the objective for this in the perspective of managing the immune response is possibly restoration of Th1, Th2 cytokine balance is one of the mechanism of establishing the homeostasis. In a layman's terminology, if you want to assess that my immune system is better than yours, so a symbolic expression is made to the size of umbrella. So here the size of umbrella is your immunity. So you can say that my immune system is better than yours, right? So this is just pictorial representation, cartoon. But what are the factors that really contributes to our immunity? First one is diet and lifestyle habit, because this is something which we regularly do. Immunity cannot be developed overnight, right? So our diet and lifestyle will determine the oxidative stress inside the body, and greater the oxidative stress, greater is the damage to our system. And to combat that, what we require is antioxidants. So antioxidants in the form of enzymatic antioxidants, which are there in the body. So this is what we are born with, or we can develop, and that is our own enzymatic system: glutathione transferase, glutathione reductase, superoxide dismutase, catalase. So these are the enzymes, enzymatic antioxidants. Whereas non-enzymatic antioxidants are the vitamins that we supplement or we derive it from our food, trace elements like zinc, selenium, and the phytochemicals. So here the role of phytochemical comes as a non-enzymatic source of antioxidants. Pre-existing illness. So nowadays people say if you are having hypertension or if you are having diabetes, then you are likely to enter into more severe form of reaction. So pre-existing illness is also a contributing factor. Genetic and epigenetic makeup. So still people are exploring this part for specifically for COVID, that do genetics play a role in this? That means if you have a specific gene polymorphism, are you more susceptible towards development of any severe condition in the treatment phase of COVID. Besides this, your sleep and wake up cycle plays a major role in deciding your immune system. So that's why the current adult crowd or the youngsters who are busy in their small gadgets of mobile, and they wake up till late night, and ultimately their sleep and wake up cycle is disturbed, and that makes you susceptible to many opportunistic infections, including viruses. So for the students, this can be a class for immune system where your immunity is mainly divided into innate and acquired. So this is what we are born with, and this is what we acquire during our lifespan. It can be natural or it can be artificial. So natural is the active infection that is currently we are facing, or this is passive when a child is born, and if the mother is infected during that phase, then the child may born with the antibodies required for fighting this infection. Artificial is vaccination. So currently what we are much more hopeful that we will get a good vaccine so that we all will get rid of future infection. But from where these cells come? So if you look at the bone marrow is the major source for our hematopoietic stem cells. And these hematopoietic stem cells, they are majorly having two subcategories of progenitor cells. That means myeloid progenitor cells and lymphoid progenitor cells. And their further downstream differentiation will generate B and T lymphocytes. And these are mainly responsible for adaptive immunity. Whereas the myeloid progenitor, which gives rise to RBC, platelet, neutrophil, eosinophil, basophil, monocytes, and these monocytes later on gives rise to dendritic cells and macrophages. So these are the innate immunity that with which we are born, and all these are associated with inflammation, and these all are actively associated with fighting with the foreign pathogens. And to keep these immune cells in circulation so that they can reach each and every organ of the body, starting from nostrils to the genitalia, through two major pathways, which we call it as highways. One is circulatory system, where blood is the carrier. One is the lymphatic system, where the lymph nodes and the lymphatic vessels and their channels are carrying all these lymphocytes and all those through these lymphatic pathways. And these are always in constant interactions, or they play a interrelationship between the circulatory system and lymphatic system. And ultimately, they makes you more immune, or they makes you more prone to infections. Right. So then the question arises: is there any way to prevent to get rid of this? And every one of you must be having some sort of preventive measures, as simple as hot water gargle or drinking hot water, to some chemotherapeutic agent for keeping yourself fit and immune, at least vitamin C, right? So if we broadly classify them into the different arms of the medical field, allopathic, Ayurvedic, and homeopathy, sorry, then in allopathy, in the initial days, SCQ was dominating, hydroxychloroquine, it was given to many, then azithromycin, the antibiotic. Later on, remdesivir came because they thought that since it is a virus, any established antiviral may work. And later on, even anti-HIV drugs were also tested and trialed on the patients with coronaviruses. And now we are aiming at vaccines because these trials have already begun. At the same time, Ayush gave a push and entered into the active chemopreventive regimen or therapeutic regimen, with various mono herbal formulations or polyherbal formulations like Sunti. So Sunti is a dried ginger powder. And there are many Ayush prescribed decoction. You must be taking all those decoctions, Kada, Okada, all those. And this is where Immunize, so that is one of our proprietary polyherbal formulation. And this has got approval from Ayush government of Gujarat for conducting clinical trials. So currently those trials are going on. And at the same time, yoga pranayama for where also propaganda was there to carry out all these as regular activity to keep yourself fit and to make your lung properly functional. So you ventilate yourself. Even there were free circulation of arsenic album along with the Ayush prescribed medicines. And many people took that as one of the prophylaxis for chemo prevention. Right? That means if you are taking any of these, how you ensure that you are protected or you are prevented from this infection. That means we should have some sort of mechanism to assess that or to validate that, right? So there is a lot of opportunity for biologists to establish a in vitro model and to establish biomarkers to validate these claims. And that's how you all biologists are aggregated here today so that we can discuss and come out with some real SOPs. Even if you look at broadly the treatment regimen still existing today, HCQ has been stopped. Remdesivir is still given, even though it is being claimed by WHO that this is inactive. Tocilizumab inactiveness is also claimed by this BIIS-7 trial. Anti-HIV drug, interferon therapy. So all these are currently going on somewhere or other, right? So one point was repurposing of drug. Repurposing means the drug was there in the market for some reason. Say HCQ was there for say arthritis or say for malaria, but they thought that this HCQ will be useful for treating this viral disease. Remdesivir was an antiviral agent for a specific viral illness, and it was thought that can this viral antiviral agent be used for this? So they tried to repurpose this drug for COVID treatment. And for this repurposing, a lot of opportunity was developed for bioinformaticians. So any of you who is interested in bioinformatics as your future career, possibly you will find in these 6 to 8 months, there are around a few thousands of papers that came into, that they are published. And all of them were having some bioinformatics analysis in that. We'll discuss that later on in this presentation. So there is a definitive role of macro and micronutrient, which is either derived from your food source or they are synthetically being supplemented as a part of the chemopreventive protocol. So there is a lot of opportunity for those who are in the field of food science and nutrition. Vaccine is, people still look skeptically towards the efficacy of this, but let's see with the initial result of the vaccine once it comes out. So there are a few failure of major therapeutics because they were not effective. So WHO recommends against the use of remdesivir in COVID-19 patients. And the tocilizumab, which is IL-6 inhibitor, and that was used to treat the cytokine storm. So the one of the largest trial, BIIS-7 trial, which had 243 patients, they found that tocilizumab was not effective in preventing intubation or death in hospitalized patients with moderate disease. Vaccine is still, we are looking as a hope, though there was a big hype in the race of vaccine development globally. So there are 20 to 30 major players who were in the race of vaccine development. And there are different types of vaccines which are being developed. These vaccines are of either the whole protein, whole capsid protein, or the whole inactivated virus, or it is nucleic acid based vaccines by Innovia or Moderna, or these are fragments of peptides which are derived from the structural protein of the virus. They also used as vaccine, or some modified viruses to trigger the immune system. So there are many different types of that, and therefore people look at with a question that how efficacy or which one is more efficacious, and what about if the virus keeps on changing their genome, or if the mutation is high, will it be still effective? And what about a long-term immunity, or every year we have to take this? So a lot of questions are there in the mind of people. Looking at all those failure of allopathic medicines or currently no clear-cut guideline for the treatment protocol, Ayush came up with many formulations which were there in our ancient literature, in the Ayurvedic literature, and they supported a lot in this phase of pandemic. So COVID-19 pandemic has posed several challenges to the Indian healthcare system, right? And in absence of proven therapies, the empirical evidence is being used for treatment of COVID-19 disease, which involves either symptomatic treatment or supportive care or prevention of complications. However, no specific drug or targeted intervention is yet available in our hand. So in the ethics context and in the interest of larger public good, inclusion of simple and safe measures from Ayush system in the integrative protocol for prophylaxis and treatment of COVID-19 should be recommended. So that's what the whole this publication from Sarika et al. They emphasized on. So they gave the example of several rashayana like ashwagandha, guduchi, amlaki, pipali, mulethi, shatavari, yastimadu as immune modulators, and this has been used since ages, thousands of years. People have used this and they protected themselves, their immune system. And there are some classical preparations. So these are individual immune modulators, whereas there are certain classical preparations which have more than one, and like Sudarshan Ganavati or Sham Shamnivati. So these are more popular in these days. There was one good commentary being published by Dr. Bhushan Patwardhan. So he's a renowned phytomedicine expert and is in the leading position in ICMR and many boards which are associated with or which are assessing the efficacy of all those phytochemicals. So one such rasayana was emphasized more, and that was ashwagandha. So if you look at the progression of SARS-CoV-2 or any viral diseases, the early response or the early stage is reduced Th1 response, increased macrophage function, and decreased natural killer cell function, and lymphopenia. So lymphopenia shows decrease in lymphocyte count when you do a complete blood count analysis. So if you look at a CBC report of a viral infected patient, the best remarkable and feature in the CBC report is lymphopenia. So the lymphocyte count goes below 20%, and you are more susceptible because all your fighting armamentarium is there in the white blood cells. Right. The next stage is the more severe stage where inflammation and all that stages comes. So immune cell infiltration, cytokine storm. So all these TNF alpha, interferon gamma, IL beta, IL2, all these cytokines, they start severe alteration in the circulation. And based on that, we will be able to diagnose that whether the patient has entered into cytokine storm or not. And if it is unmanaged or if it is not properly managed, the patient may end up in the end stage, that is acute respiratory distress syndrome, sepsis, organ failure, shock, and ultimately leads to death of the patient. So where these possible role of ashwagandha is, the supplementation of this will improve Th1 response and increased B and T cell function, increased natural cell function. And because of its antioxidant mechanism, it leads to immune homeostasis, and it improved antiviral immunity as well as it protects our lung. And since these are antioxidants, there is a much more anti-inflammatory effect of this rasayana and its constituents, and it downregulates various inflammatory mediators like COX, INOS, TNF-alpha, L-beta, L6, and therefore a reduced damage is caused. So the take-home message from his commentary was the interdisciplinary research on evidence-based Ayurveda interventions should be prioritized with an open mind for prophylaxis and add-on treatment for SARS-CoV-2 infection. So people learned lesson from China also when we all were starting or struggling with all remdesivir or SCQ or tocilizumab. They started applying the Chinese system of medicine, the complementary system of medicine, and they found good success in more than lakh. Right? So that opened up to the global community that there is a possible clear-cut role of these complementary medicine, which we call it as Ayurvedic medicine or Indian system of medicine. And we focused on that aspect. Sorry. So when we look at those natural products as phytopharmaceuticals, we name it as from farm to pharma, because farm is the place where we grow them, and pharma is the place where they are produced as medicine, and we take that. So for this farm to pharma approach, there are two major approaches. One is classical approach. The classical approach is you follow the ancient literature, get some clue based on Ayurveda Siddhanta, and that's what the Ayush guideline suggests us and gives us prescription to take. Second is you follow your instinct. So if you are vigilant enough and if you are intelligent enough and if you are observing more keenly, possibly your instinct may give you support. Or based on folklore, that means Kimbadanti. So what practices were there in Gujarati people says Dadi Manu Daitru. That means what Dadima told in their earlier days. So they were in the form of folklore, or the tribal practices still going on in various parts of the territory of India. Right? But we being the scientists, we don't believe on those, right? So what we read is we try to generate scientific evidence. So how these scientific evidences can be generated? In the current situation, it is in silico. That means whatever prediction that you do in computer using bioinformatics, based on those clue, you try to simulate them and you try to validate them in vitro. After in vitro, in vivo on various animal models, and then you try to conduct human trials. So if you look at plant metabolite as drug-like candidate, so what are these metabolites? So these are the chemical compounds that occur naturally in plant, and these are responsible for its own organoleptic property. That means taste, sight, smell, right? So if you smell a flower, it is different than the other. If you smell the leaf of a plant, it is different from the other. If you taste them, the tastes are different, right? So all these taste, sight, smell, color, all these are because of those metabolites. And Hippocrates, he was possibly the first one to prescribe the willow tree leaves that it can cure fever, right? And later on, after good pharmacological experimentation, analytical experimentation, people could find out that the active component of that willow tree leaf or the bark is rich in salicylate. And this salicylate is currently salicylic acid or aspirin. It came into market as a drug. So how from a farm to drug can be reached? This is one of the best example. That means there is a search for active metabolite. So this can be a primary metabolite or a secondary metabolite. And based on that, the source can be anything. It can be from leaf, it can be from flower, it can be from fruit, it can be from bark, it can be from root, any part of the tree. Right? So the primary and secondary metabolites are further classified into, say, small molecules or large molecules, alkaloid, terpenoid, flavonoids, glycosides, phenols, all those. And these are now being trial and tested on various targets. And these targets can be cancer, the target can be bacteria, the targets can be various viruses, fungus, right? So you will see a large number of small research is also being carried out in colleges, in universities, where the student selects a specific plant or plant part and it will try to get the extracts, aqueous extracts or solvent extracts, and he will look for antibacterial, antiviral, antifungal, anti-cancer activities. And if you are lucky enough, you may click anything good in that. But if you look at the FDA databases, right? So in the last few decades, around more than 1,500 drugs which were approved, I think 42% of them were either natural phytopharmaceuticals or natural product derivative, botanicals, or some semi-synthetic derivative of that, right? So there is a greater need of this phytopharmaceutical. So how the drug discovery from plant or from any biological substances can be initiated. So if you look at the whole drug discovery pipeline, the first phase is you should know the target, right? So here, say in terms of anti-COVID botanicals, we say virus is the target. Now, is it the whole virus, or it is the spike protein, or it is the enzyme hemagglutinase, or it is the RNA of the virus which is to be targeted? So that depends on your approach, how you are going to approach that. That means for the first step is target identification, right? So if the target is known, then there are ways like ligand-based drug discovery, structure-based drug discovery, or fragment-based drug discovery, where people will reach to a lead compound, right? So if you have, say, 1 million compound library, and if your target is known that I want to hit the spike protein, and the spike protein has got a 3D structure like this, and it has got a cleft, which is the binding site or active site. So then out of these whole lakhs of chemical repository, which molecule can go and fit with that? So using bioinformatic tools, you can identify that within a few days or few hours, depending on how big is your computational system. So if target is known, then target to lead is one aspect. Whether the disease is known. So if here the disease is virus, right? So from that disease, you will end up with the target, say spike protein, or the ACE2, which is the target, so that if your molecule goes and bind to ACE2, it will not allow the virus to bind, right? So that will be a prophylaxis. So from disease to target and from target to lead. But in all these cases, the issue is it requires a lot of time, it requires a lot of money, because ultimately you need to validate in the high throughput screening system, right? So one way is to have the ancient text reference, right? During the exam period, you must have felt that cramp that if I could get the IMP, the work would have been very easier, right? So everyone will ask for, do you have an IMP for today's exam? Similarly, for getting a right heat or for getting some clue, there is ancient text references, so that you don't have to hit and trial randomly, but you have some reference point that it has been used since thousands of years, that these phytochemicals are more useful for at least fever, for cold, for this, right? And these are in different form or preparation. Churna, that is powder. Bhashma is even the superfine powder of that. Quath is an extract of that. Ark is again the decoction part of that. Prasayana, Leha, Lepa. So these are the various forms in which this Ayurvedic system of medicine is being prepared and distributed. Fourth one is chance factor and serendipity. That means you hit something and you got that, right? So you cannot believe in that, but many a times it happens. So library versus cell line. That means you have a long list of library and you have a facility for high throughput screening. And if you're lucky enough, you may get a right hit. This is again just a pictorial representation of that. So once target is selected, your lead discovery through bioinformatics is carried out. And then the whole structure activity relationship is being done through medicinal chemistry. And then you end up with in vitro studies, followed by animal model experimentation. And finally, it enters into clinical trial in human being. And at this stage, if there is failure, then there is a big loss to the pharma industry, right? So the current approach that people have adopted for this antiviral race is they try to predict in silico first. So what they did, if they are looking at natural phytopharmaceuticals, they selected the whole list of phytochemical library that is available. They have a list of targets, right? So if you look at bioinformatic publication in these 6 months, you will come across all those targets. So here the potential targets are like ACE2, that is the receptor on our cell where the virus binds. Or it can be the spike protein, the crown of the virus, which goes and binds to ACE2. It can be hemagglutinin esterase. So ultimately that cleaves away the outer cell and allow the virus to enter into. So it is the main protease, either MPRO or CL-PRO. So based on this, there are thousands of publications, and our is also one of them. Identification of potential inhibitor of coronavirus hemagglutinin esterase using molecular docking, molecular dynamics, simulation, and binding energy. So you start with the library of compound, you have your target, then you sort out a few tens of these molecules. Then the first filter is Lipinski rule of 5. That means can it act as a good drug? Then ADME. ADME says absorption, distribution, metabolism, and excretion. So if it passes through this ADME filter, then you can further go and look for toxicity. So if it is not causing any organ toxicity, then you can move ahead as a good drug-like candidate. So again, with the help of molecular dynamic simulation, you can exactly see how best and how fit this ligand and receptor are binding. And the whole scoring is done through the binding free energy calculation. And you end up with few molecules. So this is the in silico approach, and there are lots of publication in this and pertaining to COVID only, right? So even people have find out giloy as one of the potent herb, and the phytopharmaceutical present in the gilloy were screened against the viral targets. So once you end up with few number of phytopharmaceutical, the question arises that is it really antiviral, right? So people come up with a claim that my formulation is 100% antiviral. Even Baba Ramdev also claimed that, and later on it entered into controversy. But before entering into that, possibly you have to go through at least some in vitro assays. In the initial days, there were no in vitro assays which were available for confirmation, because to handle this virus, you require biosafety level 3 facility, right? And many of the labs or the pharma company even don't have this biosafety level 3 facility, which can handle the viruses, the highly infectious and pathogenic viruses. But if you have that facility, possibly we can have the in vitro assessment. So in which what they do, the target cell. So usually people use the target cell which are expressing ACE2, so that your virus spike protein can go and bind to that and get entry into that. But Verocell is one of the most ideal in vitro cell line system. And when these cells are incubated with equal exposure of viral load, in one, they don't put any substitute like any phytochemicals, and in the other, so that acts as a control, and in the other they use along with the phytochemicals. And they try to see that by addition of phytochemical, does it restrict the entry of virus into the cell? And if it restricts the entry into the cell, the total number of virus after 24, 36, or 48 or 72 hours of exposure, there will not be increase in the viral copy number, because virus cannot replicate outside the human cell. So that control shows that if it is not getting entry, what is the total number, and if it gets enter into that, what will be the number? So based on that and the color intensity, so it says, so it is a luciferase based assay where the number of living cells, so luciferase activity in drug treated cell versus luciferase activity in untreated cell, will give the inhibition of viral replication. So whether you are preventing the entry of virus, whether you are preventing the replication of a virus, that also can be done through these in vitro assays. There are other publications which shows that since we know that the entry of virus is required for its replication. So if the virus enter into that and if it replicates, its total genomic copy number will also increase. So instead of a color development, you can also use a RT-PCR based assessment where you can see that there is an increase in copy number of virus. So that means they are replicating. So if there is inhibition, it will not replicate at all, right? So based on that, you can predict whether your compound is antiviral effective or not. There are various in vivo models which are recently being trial and tested, because these were not available earlier. People initially looked at mouse models because those are the poor animal which are under our control. But they found that they lack appropriate receptor to initiate viral infection. So a normal mouse will not have ACE2, which is compatible to the spike protein of virus. So what they did, they tried to mutate the spike protein so that they can enter into, or they can bind to this ACE2 receptor, or they adapted this ACE2 receptor for the spike protein, or this ACE2 are genetically modified, and the humanized ACE2 is being incorporated into the genome, and those genetically modified mouse were used as a mouse model. Even people used cereal, hamster, ferret, monkeys, mink, cat as the mouse, the in vivo models. But Is there any other way out, right? So the classical approach and for direct supplementation. So if someone says that go and take ashwagandha, you can blindly go and take. You can take turmeric, right? Because all these are falling under GRAS category, GRAS, that is generally recognized as safe. And that's why they don't require any further prescription or recommendation, so that they can be taken with direct human supplementation. And many of the Ayush prescribed medicines like Samshamnivati or any Quath are being supplemented to human being, and studies are being carried out. But if I come up with innovative plant source or a novel source whose uh, representation is not being mentioned anywhere in the literature earlier, then it makes an issue. Then you have to pass through the whole series of in vitro, in vivo, all those, right? So those are novel source. And what can be done for that? So we did develop some simulated digestion approach model, and I will show you that. So the direct supplementation is one of the example is Immunize. So Immunize is a proprietary polyherbal formulation. It was approved by Ayush government of Gujarat for conducting trial on asymptomatic, mild symptomatic patients, and after ethics approval. So we got all these approvals, and we have started the Immunize trial in patients with mild and asymptomatic cases. So the dose was 4 tablets twice a day for 14 days. And the study design involves for monitoring of vitals, that what is their oxygen saturation, fever, any change in symptom, right? So all these are fed into a questionnaire regarding their symptoms and other medications which are supplemented with this. They were tested for CBC, CRP, D-dimer, LFT, RFT at day 0 and after completion of 14 days trials on day 15. And they were also assessed on day 28 to check whether they have developed antibody or not. So this was the simplest trial design that was conducted. Now, how the innovator, so with this government of Gujarat University was one of the research partner, and one Ayurvedic doctor, Dr. Rakesh Rawal, who was the innovator of Immunize. He partnered with us for further development of this project. So I can say Immunize was a repurposed drug because he was using Immunize since last 10 years. So for patients with various autoimmune diseases, for various immune dysfunction, including HIV, right, autoimmune hepatitis, all those cases, he was getting good results. So that prompted him to speculate that if it is really working in immune modulation, why it can't be used for patients with coronavirus infection. And that's how, with all prior evidences and prior reports in animal models and its toxicity studies, finally we got approval from Ayush. And for unknown substance from unknown origin, the best way is what we have proposed, that you prepare the plant extract, right? Whatever the source may be, leaf, flower, root, shoot, fruit, whatever it is. You prepare the extract, whether it is aqueous or organic solvent. Now, synthetic salivary fluid, synthetic gastric juice, and synthetic intestinal fluid. So these cocktails are available, the composition is available. So what we are doing, we are mimicking as if it is being ingested by a human being. So first of all, it will go to our mouth cavity where it will interact with salivary secretions. From that, it will go to gastric phase, that is stomach, where it will interact with gastric juice. All these mixture will go to intestine, and where it will interact with the intestinal fluid it will get a long interaction with gut microflora. So around 15 to 20 hours of incubation is there. And after this gut microflora incubation, whatever supernatant that you get, these are passed through a Keiko2 monolayer insert. So this mimics the intestinal absorption. So if you put everything on the top, it passes down through this Keiko2 monolayer. So that this mimics the intestinal membrane. And at the bottom, there are hepatocytes. And these hepatocytes will mimic the liver transformation. So biotransformation that is being taken in the liver, so it will mimic that. So if you take something in a natural way, it passes through all these phases: salivary, gastric, intestinal, gut, and then it gets absorbed into blood, and that goes to liver. And then whatever biotransformation happens, the metabolites are generated out of these will be in circulation, and they will reach the target cell. So then whatever supernatant that is being taken from this after hepatocyte digestion will be used for screening in the in vitro model, which was shown earlier. And then we can say that really this is the phytopharmaceutical, and these are the metabolites which are acting on the target, and that is effective. The third innovative approach that we are currently dealing is we all know that we are what we eat, right? And there is a current group of people, vegan group. So what they are doing, they eat raw, whatever is there, right? So any vegetable, any fruit, any this, unprocessed. So eat raw vegan approach. So what was so beautiful about this, right? So what we did, we proposed a project with 3 antiviral plants. One is Osimum, Curcuma longa, and eucalyptus. So what we did the sequencing of the whole genome of all these plant sources. Currently, Osimum is done, Curcuma longa and rest of the antiviral plant sequencing is going on. So our Osimum basilicum sequencing that revealed that there are few microRNA which are reported in or which are there in the Osimum basilicum, and these microRNA, they have got the exact complementary sequence of the viral genome for the COVID. So there are 4 different targets of these COVID viral RNA, which can be targeted with the Tulsi microRNA. That means if you take the raw Tulsi leaves, say 10 to 20 leaves, you chew them up and then you engulf, and the rest of the digestion and all those process goes on. And these microRNAs are being trapped into the vesicles, and they bypass this all digestive processes. And when they enter or they passes through the gut and enters into circulation and ultimately reaches the destination cells, they can act as the antiviral agents. So this was one of the hypotheses. It's in vitro validation, and all those experiments are currently going on. So possibly this is my last slide. So that shows do's and don't from my side. That do exercise or yoga regularly, keep yourself fit physically, and self-hygiene is required for proper washing and cleansing yourself with social distancing. Balanced diet with macro and micronutrients, and maintain a proper sleep-wake cycles of your circadian rhythm. And please don't take medicines prescribed on internet without medical prescription or medical guidance. So avoid self-medication. Thank you. And I welcome you all to visit Gujarat University. Thank you.
Thank you, sir. Students, now we are open for questions. If you have any question or query, you can ask.
Hello, sir.
Yeah.
Good afternoon, sir. Sir, Sir, you told us apart from the coronavirus entry point and the exit point.
Yeah.
So I understood RNA virus binding on the ACE2 receptor.
Yeah.
That is our DNA receptor. So, sir, I want to know about the exit point, sir. How coronavirus RNA exit to our body, sir.
So if I go to that slide again, yeah. So here, after entry, all the structural protein and all those are lost, and the whole RNA, right, that is reverse transcribed, and again the multiple copies of that are made from RNA polymerase. So say a single entry of virus may lead to some thousands of or lakhs of copies of these RNA. Along with that, viral structural proteins and all those which are coded by different genes are also made. And these are packaged in our Golgi. And all these thousands or millions of these viral particles, they are then released once the cell, either the cell bursts or they are being thrown out through exocytosis. So these are all packaged virus, an exact copy of the virus which was there and which entered into the cell.
Okay, thank you, sir. And sir, a similar another question is mine. Sir, can sir elaborate about the adaptation of ACE2 receptor in mouse, sir, for trial on the medicine in the mouse, sir?
So as I told, the initial ACE2, which is there in the mouse, that was not of the similar conformation like the human ACE2, right? So when they exposed these mouse with a cocktail of these viruses, and when a cocktail of this virus, there are many variants within these viruses. And these viruses, when they are coming in contact with this, there is a chance that some of these virus, right? So these are not exactly similar. So there are some mutations. So some of these viruses have a spike protein which is mutated slightly, and that may fit into this. So out of a million, possibly few tens of such copies can bind to this. And whatever viruses they can bind to this, they can get the entry. And then you have millions of copies of that virus. So by trial and error methods, people could succeed that a few of the mutated virus can get entry into this. And that says that you have to adapt to this existing ACE2 and whatever virus that makes entry into that, then you have got a pool of those viruses. So it's a trial and error method. Yeah.
Okay, thank you, sir. And sir, my last question.
Yeah.
Sir, is the right, sir, Ayurvedic medicine as well as Siddha medicine is more effective compared to allopathic medicine, sir, till today also, sir?
Look, the question may lead to some controversy if I clearly say yes or no to something. But in the present observation, what we have seen is majority of allopathic medicine which were initially prescribed or which were initially trial and tested, because there was no clear-cut medicine which was available, because this was a new disease, right? So they tried with all repurposing of drug, and in many cases we failed, except the dexamethasone or tocilizumab, where severity of the disease can be managed. If you look at prophylaxis, there is no medicine in allopathy except the vitamin C and vitamin D, which people supplement, and that is for general maintenance of immune cell. But in case of Ayurveda, it's a long list of suggested chemopreventive or prophylactic supplements which are there and which has been tested since ages, hundreds of years, thousands of years. So that's why we can say that, yeah, chemoprevention is there and it is reported in Ayurvedic literature, but it is not there in allopathy. But once you enter into any complication in acute phase, the doctors will never opt for any Ayurvedic system of medicine, because in that case, management of respiratory distress or cardiac function is required allopathically.
Okay, thank you, sir.
Good evening, sir.
Yeah.
So I had a question. So I have been doing in silico work myself on COVID itself. So, sir, I was able to do docking and dynamics, but you just told about ADME and toxicity. So I was wondering if you can tell me which softwares or which web tools could be used for this, and what are the procedures to do that?
So there are many proprietary tools like Schrodinger and all those have their built-in capabilities of filtering. And there are many sites, online sites, where ADMET scoring and all those can be done. So possibly I can share it to the organizer after a few minutes so that she can share with you all the participants. So there are a list of such free tools are also available. So your main intention is, sorry, the ADME will tell you that which size of molecule can enter into the cell, and if it enters, how they are being distributed, and after metabolism, whether they can be excreted or not. And based on the past literature on the drugs which have developed various toxicities, so ADMET, T is the toxicity. So based on those literature, even I have one publication on that, predicting cardiovascular and hepatogenic toxicity using mathematical model. So based on the earlier literature, so FDA has listed a list of drugs which are cardiotoxic, which are hepatotoxic, which are nephrotoxic, and they are withdrawn. And if you analyze their structure to the structure activity relationship, the SARS, you will be able to construct that if there are structures with this specific type of pharmacophores in them, then they are potentially toxic to these organs. But the normal predictors which are there available online, they usually look at the different pharmacophores, and based on the pharmacophore scoring function, they will tell you whether these drugs are going to be useful or not, or a drug-like candidate. So online ADMET tools are there.
Okay, sir. Okay. Sir, second question. So I have heard about QSAR studies as well.
Yeah.
So, sir, can you please throw some light on that?
QSAR, it requires a bit of chemistry knowledge. So either if you are a pharmacist or if you are a chemical science student, then you will be able to do QSAR. So QSAR, what it shows that if this is the structure you are starting with, and there are possibilities of adding many functional groups to that, and if you keep on adding those functional group, and you try to check, so by changing the structure, how the function is being affected, quantitative structure activity relationship. So when you have a list of some compounds which are the active pharmacophores, based on that you can develop a QSAR model. And if you can intuitively find the potential sites where you can do some modifications, then you can develop the synthetic derivatives of a single backbone. Why this is required is that, say, there is a natural compound called allicin or capsaicin, right? So if you go with that in the market, you cannot patent it because it is naturally available. Now your intention is to make it more effective, as well as you can make it synthetically or semi-synthetic derivative of that. So what you will do, you will try to modify some bonds in that, and you will get a derivative which is at least equal or better than the backbone or the original molecule. Then that compound can be patentable, right? And unless it is patentable, it is not marketable. So pharma company will not look into those, right? So QSAR has got importance in that for designing or redesigning or for optimizing a known backbone for the development of drug.
Okay, sir. Okay, thank you very much, sir. So can you please send those links as well as
Yeah.
your publication?
Yeah.
I'll be glad to read it.
Okay. Thank you all.
Yes, sir, there is one question in chat
Yeah.
box, that is, since various medicinal plants like osimum and curcuma has antiviral properties, why they are not used to cure coronavirus?
So as I told, unless you develop that proof of concept. So what is currently the challenge was we don't have a good biosafety 3 facility across India, right? Very few limited virology centers or big pharma will have that. And to prove anything antiviral, we need all those facility. So unless we prove that, we cannot circulate them in the market as antiviral. Otherwise, there will be immediate claim from DCGI or ICMR against that. But currently, people are treating with all those phytochemicals, giloy, curcumin, ashwagandha, shatavari, so cocktail of that. And even our formulation is also based on that, and we are getting good results. So they can be developed, but we need some good proof of concept based on in vitro and in vivo experimentation.
So, sir, there is another question. When the student is going through some article on COVID-19, many of them mentioned that researchers are focusing on development of an effective drug targeting to prevent the contact between N terminal of N protein and a single protein DNA strand, which can stop viral replication and transcription. How actually this works? I mean, the mechanism behind that.
So as I told, all these red blocking sign, these are the point where we can intervene, right? So if the viral RNA is entering into a replicative cycle, then there are intermediate enzymes which are involved in that. So if we don't allow, or if we don't wish to allow it to replicate further, we need to target those enzymes which are involved in its replication. It can be a ribonuclease or it can be a helicase. So there are different points. So if they are they have developed their copies, then the second point is we don't allow them to enter into a combination with the nucleocapsid protein for packaging. So you can restrict them at packaging. So if you can restrict them at packaging, it will not release the active virions in the lytic cycle. So there are all possibilities starting from entry or binding of ACE2 up to packaging and release of viruses. So for that, we need to know the exact mechanism and exact molecules which are involved in the specific stages. And based on that, based on the structural, complete 3D structural information of all those intermediates or the targets, we can definitely design a drug from our repertoire.
Okay, so I think there are no more questions. And if you have any, you can drop the email or in WhatsApp group. You can ask. I'll, contact a professor for this particular questions or query. So, sir, it was really wonderful and very informative, interactive session. Thank you so much for your time. And it will going to benefit the students who are actually working right now on COVID-19 projects, and later also they have to work on some grassroots practices which are antiviral, and there are some other practices which is associated with this medicinal plant. So thank you so much, sir, and we always look forward for your support and guidance for such future workshop.
Wish you good luck.
Thank you, sir.
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 So you can note down my email ID, Rakesh M Rawal, RAKESHM RAWAL @gmail.com.
I'll send them, sir.
Yeah, you can share with them. Yeah.
Yeah.
Thank you.
Thank you, sir.
Bye-bye.
Thank you, sir.
Thank you.
Rakesh. Okay, students. So I hope you have enjoyed the session.
Yes, ma'am.
Yes, ma'am.
Okay. So here I'm sharing email ID of professor, and you can send email or you can contact him for your query. Please note down the email ID.